So today we are gonna talk about chapter 7, which was all about protein function and how that's related to structure. And we did that with 3 different examples. So first we're gonna start with oxygen storage and transport. And then later, if we have time, we'll get to muscle contraction and immune response. By the end of the week, hopefully we'll have covered all 3 of these things. So let's start with the oxygen storage and transport. So this is all about myoglobin and hemoglobin, carrying oxygen through your bloodstream, delivering it to tissues, all that good stuff. So I want everybody to take maybe 10 minutes, start thinking about oxygen transport, start filling in this table. Feel free to work with a neighbor if you'd like, and we'll go over it together in a little bit. Yeah. Yeah. So yeah, it's small, right? Yeah. So yeah, so where we did the roach alpha data show but when we start combining those together is where you get tertiary. So like for a beta airplane, you have like beta G connected by so like for so like that's what makes it tertiary because you have like beta plus. Oh, like combination. Yeah. So like, I mean, it's also true that like you can't just have like a beta G without those connected on them, so it's kind of weird, but that's how. Okay. Yeah, it does. Yeah. Really? I really don't mind. Like I tend to I've worked 2:30 and I usually start 4:30. All right, so let's start going over this table. So let's start with the function. So does somebody want to tell me the function of myoglobin or hemoglobin? Yeah. Which one? So yeah, so myoglobin binds oxygen at the muscles. So if it's binding oxygen in your muscle, it's also essentially helping oxygen to penetrate in your tissues. So when you're using up a lot of oxygen during cellular respiration, myoglobin is there to supply that oxygen locally at your muscle where it needs a lot of energy. It's also really important for oxygen storage, so you have the oxygen there ready for when you need it. So, how about hemoglobin? Yeah. Yeah, exactly. So, hemoglobin binds oxygen. So you said it binds it in the lungs, which is true. That's where it picks it up. But it's sort of, yeah, I guess. So it binds O2 in the lungs. But its job is really to then deliver it, to bring it throughout the bloodstream and deliver it where it's needed throughout the body. So those are their two functions, and they're distinct, but they help each other out, right? So we have hemoglobin picking up the oxygen after we breathe, and then carrying it through our blood, and then once we get to muscle or tissue that's oxygen starved, the hemoglobin hands off the oxygen to myoglobin, which hangs onto it until it's needed at local sites throughout your body. And so those are their two functions. And the functions, as we are trying to emphasize in this whole chapter, are helped determine or like related to their structure. So structure determines function. Function is related to structure. So like that's kind of the point of the table in particular, to help us kind of start thinking about what are the differences in structure in these two proteins and how does that give them their distinct but similar functions. So who wants to then start us off with structure? What is the secondary structure that we see in myoglobin and hemoglobin? Yeah. Alpha helix. Yeah. So they both have alpha helix character. So they have very similar secondary structures. They are both primarily made of alpha helix. So there must be other differences in structure that help give them these different functions, right? So let's talk about some of these other parts of structure that we see differences in. So can somebody tell me the number of subunits? Yeah. Yeah, exactly. So hemoglobin has 4 subunits. Myoglobin only has 1. And so that helps relate to other features of particularly hemoglobin that we'll talk a lot about as we go through these practice questions that I won't give away now. So what is another name for like a protein that only has 1 subunit, like ending in mer? Yeah, monomer. Yep. And what about hemoglobin? What kind of... Yeah, it's a tetramer. Yeah. And is it a homotetramer or a heterotetramer? It's a heterotetramer. Excellent. So it's a heterotetramer because we don't have 4 identical subunits, right? Does anybody remember what we call the types of subunits? Yeah. Okay. Did you want to answer a different question? Yeah. What do we call the types of subunits that we see in hemoglobin? Like the names of them? They have Greek letters. They don't really have names, but yeah. Exactly, yeah. So we'll often see hemoglobin written as alpha 2, beta 2. It has 2 subunits that are alpha and 2 subunits that are beta. This has nothing to do with their secondary structures. We just talked about how they all have alpha helix as their secondary structure. There's just 2 kinds of very slightly different subunits, and we call them alpha and beta just to confuse you for the most part. All right, so that brings us to a related question, which is how many heme groups do we see in each of these proteins? Yeah. Exactly, yeah. So there is 1 heme group in each subunit. So myoglobin, 1 subunit, 1 heme group. Hemoglobin, 4 subunits. Each subunit has its own heme group. We've got 4 heme groups per hemoglobin. So, so let's start to talk about their binding curves. Does anybody remember the name for the shape of either of those binding curves? Yeah. Mm-hmm. Yeah, excellent. So myoglobin has a hyperbolic binding curve. I will try to draw one without hopefully destroying this, but it approximately looks like that, right? And then hemoglobin has a sigmoidal curve. So a sigmoidal, hopefully you should start thinking S shape, sigma S. So hemoglobin's curve is a little bit more like this. So it has a sort of S shape, especially towards the left, and then towards the right, it looks a little bit more like the myoglobin curve does. So why do we get those different shapes of those binding curves? What type of binding do we see for each of these proteins? There's a special name for it, yeah. Exactly, yeah. So which one has cooperative binding? Yeah, exactly. So hemoglobin has cooperative binding. And we'll talk more about how that ends up giving it a more S-shape binding curve. So essentially we have kind of a slower rate of binding given oxygen pressure at the beginning, and then as we have increased oxygen, we have more and more, it's better and better at binding as we get more binding. So that was a really terrible way to say it. So let's say that another way. So for cooperative binding, once 1 subunit of hemoglobin binds oxygen, that makes the other ones more likely to bind oxygen. And then if you add on a second one that binds oxygen, that makes the other 2 more likely. And if you add on a third one, that makes the 4th one more likely. So at the beginning here, if you have only 1 subunit bound, it's not super likely. But the more subunits that you get bound as you go up this curve in the y-axis, the easier it is to get oxygen in there, so the curve gets steeper as you go. And that is in contrast to myoglobin. Myoglobin only has 1 subunit. There's nothing to cooperate with, right? So it's non-cooperative. There's just, either the heme group has an oxygen in it or it doesn't. It's not talking to any other subunits, is not connected to anything else. And so that's why we have this relatively sort of simple hyperbolic kind of binding curve for hemoglobin. Any questions so far about the differences and similarities between myoglobin and hemoglobin? Yeah.
Would you say the cooperative binding as 1 oxygen binds, it increases the binding affinity of the groups next to it?
Essentially, yeah. So we're going to get more into it, but there's 2 different states that each of the subunits can be in that affect the shape and that affect the likelihood or the ease of like oxygen binding to the heme group. And so, yeah, I guess you could say that the binding affinity, I don't know if the binding affinity itself changes, because that's like a really technical term that I think is related probably to like the dissociation constant, which doesn't change. But it is true that they're more and less likely to bind oxygen once you get one subunit found. There's a non-answer for you. Any other questions? Okay, so let's try to get some answers out of here. So let's look at these two binding curves. So we just talked a little bit about the shapes of each one. I may have given the answer away here, but take a couple of minutes, everybody, and look at these two curves, figure out which one is which, figure out the KDs of each curve, and then we'll talk more about why they have different shapes.
KDs and millimeters of mercury, right? Or torr?
Yeah, either one. They're both pressure. Yeah, so this particular graph has millimeters of Hg. But yeah, we see torr a lot.
Are torr and millimeters of mercury interchangeable? Yeah.
I used to know that, and I don't remember, and I forgot to look it up before class. I can look that up later and tell you. Sorry. It's like something I haven't thought about in like 15 years. That's fine. Yeah, so torr and millimeters of mercury are equivalent. I thought so, but didn't want to say so without consulting Google. So there we go. All right, so who wants to tell me which curve represents myoglobin? Yeah, the green one. Yeah, that one is myoglobin, making the other one hemoglobin. So yeah, who wants to take a guess at the KD of myoglobin? The scale is not very accurate, so you can just do your best guess here. Yeah, it's about 5-ish. Yeah, it's a little lower than that, but hard to tell from this graph. How did you figure that out? What? It was in the video. Yeah. Does anybody have an idea how to figure it out? Yeah. Yeah. Yeah, exactly. So the KD is related to the fractional saturation. So on the y-axis, we have oxygen saturation percentage, which we also call fractional saturation, which we sometimes write as like Y oxygen. And so at 50% saturation, which is about here, we can draw a horizontal line over to our curve and then drop a vertical line down. So, and then the pressure at which you get 50% fractional saturation is the KD. So for myoglobin, it's about 2 or 3. It's a little under 5. And what else did I want to say about that? Oh, does anybody know another term for the KD? I did not say this in the video, but you should know it. So that's why we're just wondering if anybody else has another word for it. No. So another word for the KD is the P50. So KD or P50. So the P50 refers to the pressure of oxygen that yields 50% binding. So that's why it's P50, right? And that's the same thing as the KD. The KD being sort of the pressure at which you get 50% dissociation, or which is also the same as 50% binding. So when you're halfway full, you are at the KD or the P50 in terms of how much oxygen you have in the environment, which is on your x-axis. So for myoglobin, it's about 2 or 3. What is it for hemoglobin? Yeah, it's a little, again, it's hard to see with this graph, but yeah, it's a little bit under 30. So if we extend over our 50% over to our hemoglobin curve, and then we drop down a vertical, it's somewhere around, maybe 22 to 25. And as we also we'll talk about and have you saw in the video, this curve shifts depending on like the pH of the environment and like other factors. So this is sort of approximate. That's why we have like ranges here, especially for hemoglobin. So which one has a lower KD? Not a trick question. Yeah, myoglobin. So what does that mean in terms of how strongly it binds oxygen? If the dissociation constant is lower, is it binding more strongly or more weakly? Yeah, more strongly, yeah. So if you have stronger binding, you have less dissociation, meaning you'd have a lower KD, right? Make sense? Cool. All right. So yeah, let's talk a little bit more, unless somebody has more questions about KDs, we can talk about why do the two curves have, you have a question? Yeah.
So when it comes to KD, we're always referring to the 50% mark.
Yes, the 50% fractional saturation will always get you the KD.
And then is it like a question-on-question basis, or is myoglobin usually the binds more strongly compared to hemoglobin?
Yeah, well, I guess it depends on where you are in terms of oxygen pressure, right? So if you're looking at, pressure of 20 torr, 20 millimeters of mercury, myoglobin is going to bind more strongly, right? We are at like 90% bound here compared to like a little under 50% bound for hemoglobin. So more bound means it's going to grab it on tighter, right? But if we're looking like over here at 100 torr or 100 millimeters of mercury, they're about the same. So depends on the environment. But yeah, in general, you could say myoglobin is almost always going to bind more strongly because you see it by the curve. You see that it's above the curve for hemoglobin. Yeah. Yeah, so the KD refers to dissociation. So just like with any sort of K value, we have products in the top of the fraction, reactants in the bottom. So at the top of our fraction, we have separate protein plus oxygen. In the denominator of our fraction, we have bound hemoglobin or myoglobin. So unbound is on top. So a smaller fraction would mean less is unbound, right? And so more is bound, meaning it's stronger binding. Sure. So like the KD basically it's essentially a fraction that's unbound over bound, either hemoglobin or myoglobin. So if the numerator is smaller, meaning the KD is smaller, that means you have a larger denominator, meaning more is bound, because you have stronger binding. Okay. It's like, yeah, it's one of those things you got to think through. I mean, you can also think of it as how much oxygen is required to get 50% binding. So, like, you only need a really small amount of oxygen to get half of your myoglobin bound. You need a lot more oxygen to get half of that hemoglobin bound. So, myoglobin will grab on more strongly to the oxygen. It needs very little oxygen in the environment to bind as well as hemoglobin would. If that's another way to think about it. Okay. Depends on which axis of your graph you like. Any other questions about KD? Okay, so let's talk a little bit more about why the two curves have different shapes. So we kind of talked about this a little bit on the last slide, but I think it helps to think about it again while we're actually looking at the curves. So we know that there is cooperativity, right? So hemoglobin has cooperative binding because it has 4 different subunits that can bind. So if you look at sort of I'm just going to erase the KDs so we can look at this curve. Easier said than done. So if you look at the bottom part of hemoglobin's binding curve towards the left, it's relatively shallow. And if it's helpful, you can almost think of this as it's kind of the beginning of like a very shallow hyperbolic curve. So like if hemoglobin only had one subunit with no cooperativity, it would resemble kind of like, this is not a great curve, but you get the idea of like it's overall shallower. And so like there's not super strong binding, you never get to full like saturation. But as we know, like once one subunit of hemoglobin binds an oxygen, it makes the other ones more likely to bind. They want to bind. And so the curve will start to get steeper and resemble this. It'll eventually, like once you get over here towards the right, where more and more subunits are bound, it looks more like the hyperbolic curve for myoglobin. So in a sense, like you have like the nature of one hyperbolic curve on the left, you have like a different kind of hyperbolic curve on the right, and when you kind of smush them together, you end up with this kind of sigmoidal curve, where the slope gets steeper, the more oxygens are bound, the more subunits have an oxygen in each one, if that is helpful to think about. And so another way to think about it is less increase in oxygen, partial pressure of oxygen is necessary. The more subunits are bound, the less increase in oxygen you need to add oxygen to your next subunit. So like at first it's kind of difficult, and then once you get over here, like the difference in oxygen you need is relatively small for like those rays on the y-axis, if that's helpful to think about. So does anybody have questions about cooperativity, KDs? Nope. Okay. Let's think about oxygen binding. So here is another question. So blood normally contains between 30 and 100 torr of oxygen. And please refer to the graph on the last slide when you're answering these. But I'll give everybody another 5 or 10 minutes and think about what is the fractional saturation of myoglobin and hemoglobin over that range of oxygen pressure in the blood, and how does that relate to their different biological functions. So I'll give everybody about 5 or 10 minutes for that. you want to get us started? Yeah, sure.
Why is the range so wide for pressure in the blood?
Because there are a number of different environments in your body, and oxygen varies a lot, right? So, like in, let's say, your lungs, a lot of oxygen, right? You're breathing in, you're taking in oxygen, lots of oxygen there. Let's say you're like exercising and your muscles are working really hard and they're using up oxygen to create energy. There might be a very low concentration of oxygen, like in that particular location. Even though you're breathing in air at your lungs, it's very high. That's why we have the system of hemoglobin and myoglobin to take the oxygen where it's coming in at your lungs and bring it to the rest of your body where you can't breathe over there, right? So you need somewhere to get the oxygen to those other places, especially as you use it up and deplete it during cellular respiration. All right, so with that, let's start talking about this question. So what is the fractional saturation of myoglobin over this range of 30 to 100 torr? So let's look at this graph. So if we have about 30 torr, we can drop a line around here. 100 torr, they already have a line for us. So what is our fractional saturation of myoglobin over that range? Yeah.
Around 97, 98.
Yeah, exactly. So like we start around here, maybe 95-ish, we get up to close to 100. So it's something like 95 to 100%. Again, that's a little bit hard to get exact with that graph because there aren't really good gradations between the numbers, but around there. So let's look at hemoglobin. What do we see for hemoglobin over that range? Yeah. Yeah, so I would say it's probably closer to just about 60 there, but yeah. So yeah, it's about 60 to 95 or 100%. Let's just say 100% to make it nice and round. But this is approximate. So the fact that we have these differences in fractional saturation over this range of oxygen that we see in the blood, what does this mean for the different biological functions of myoglobin and hemoglobin? Yeah. Go for it.
Since myoglobin has to store that energy, it has to come down to it much tighter than hemoglobin, which is more of a transporter, and therefore has a low or a wider range of fractional.
Yeah, that's a good way to start. So it kind of comes back to Juan's question before about where are we seeing these different amounts of oxygen in our body. So at our lungs, where we have a lot of oxygen, that's where hemoglobin is going to come in and pick up oxygen and start carrying it around. So we want hemoglobin to have a pretty high fractional saturation, like when it's at the lungs. But once we're now traveling around in the blood to the tissue, hemoglobin has to then drop off that blood where it's, or not blood, it has to drop off the oxygen where it's most needed. And that's where we have the lowest amount of oxygen. So when we're down to like 30 torr, now we see how hemoglobin's only about 60% saturated versus myoglobin, which really still wants to hold on to the oxygen, even at that low pressure. So that's where hemoglobin can essentially hand off the oxygen to a myoglobin that's going to hold on to it at the tissue. And that's basically hemoglobin's job, right? It's taking oxygen from the high oxygen environment, bringing it to a low oxygen environment where it no longer wants to hold on to it very tightly, and that allows it to then deliver the oxygen to the myoglobin, which holds on to it and gives it to lets it penetrate deeper into your tissue, for example, at your muscle. So Okay, any questions about this concept of different fractional saturation at different oxygen pressures? Yeah.
Might not be like super topic, but like, how does the myofilament move through the tissue?
How does it move through the tissue? I don't know actually. I don't know that it necessarily moves through tissue. It might just be. I honestly have no idea. That's an interesting question. I can look into that and get back to you. Yeah. Any other questions that I may or may not be able to answer? All right, so now let's talk more about that cooperativity. So we already know that hemoglobin has this concept of cooperativity between its subunits or among its subunits, and that is related to when the subunits are in the T state and the R state. So everybody take a few minutes and look through these, see what you remember, and then we will talk about it together. All right, so I want to actually go over these a little bit out of order because I think it will hopefully make everything make more sense at the end. So I actually want to start with number 2. So which state binds oxygen more strongly, the R or the T? Yeah, the R state. Yes, that is correct. So Yeah, and that switch from the T to the R state, again, is what is responsible for the cooperativity that hemoglobin has. So if everybody is in the T state, which is fairly loose, and then once an oxygen bind, we switch over to the R state, and that allows the other neighboring subunits to also want to adopt the R state, which makes them more likely to bind oxygen, ultimately helping all 4 subunits want to bind oxygen more strongly. And so I'm actually going to skip over now to number 6, because having the shape of the heme group is one of the more direct causes of what helps shift the whole subunit's shape from the T to the R state. So does anybody know which state has a planar heme group? I don't think I said this in the video, so nobody might know this, but you can guess. Yeah. It is the R state, yeah. So, yeah, so if you recall, the heme group has this porphyrin ring structure, and it can either be like very flat and planar, or it can be not planar. I can't remember the word for that, I guess. But yeah, it's it can be more sort of like angular, or it can be relatively flat. And so that shift from having distinct angles, like a sort of squishier ring versus like a flat planar ring, will help to then will have an effect on the surrounding amino acids that are part of the actual protein subunit, and ultimately help that whole protein subunit adopt that T state shape or the R state shape. So planar R state. Okay, so, all right, so then let's talk about now which one of the T state or R state has a larger central cavity. Yeah, the T state, yeah. So the T state has a larger central cavity. And what goes in that central cavity? Yeah. No, sorry, that is not where the iron goes. In the actual, so like that's in the actual heme group, which is sort of more like central within each subunit. So this is like the central cavity of hemoglobin. It has like between all of the subunits, kind of. Anybody have any other ideas for what goes in that central cavity? Yeah.
I don't remember the name, but it's phospho something glycerol?
Yes, yeah, so it's BPG, which is question number 4. So, yeah, so the larger central cavity is what we see in the T state. So in which state do we see where we have hemoglobin binding to BPG? Yeah, the T state, exactly. So the BPG fits in that larger central cavity when it's in the T state. When BPG binds there, it stabilizes hemoglobin into that T state, because BPG kind of like parks itself in there, and it gets kind of stuck that way, in some sense. Versus when you're in the R state, you have a smaller central cavity. BPG doesn't fit in there. So, all right. So, yeah. Exactly, yeah. So when BPG is bound, hemoglobin is kind of stuck in the T state, and that's going to encourage it to release oxygen. And BPG is a product of metabolism, so this makes sense, right? As you're going through metabolism, you're consuming oxygen. This is a time when you'd want to encourage hemoglobin to release that oxygen, so you can then use it to make more energy, because BPG is a sign that you're already trying to use energy, or you need energy, you're trying to make energy. Any questions about that? Okay, so let's then talk about which state, for number 5, which state is stabilized by salt bridges and N-terminal carbamates? Yeah, the T state, yeah. So the T state, yeah, it is stabilized by salt bridges and N-terminal carbamates. So you're helping different pieces of the subunit stick to each other. They're stabilized by these ionic interactions. And these ionic interactions, especially with the salt bridges, you you tend to have those those amino acids in a charged state at a low pH when you have free protons in solution. So having protons in solution helps to make these salt bridges, which helps form a T state. So then that can help us answer question number 3. Which structure would then be preferred at a high pH? If this low pH with protons helps stabilize the T state, what is our high pH going to help stabilize? Yeah, the R state. Excellent. And so, yeah, this is sort of, hopefully this kind of illustrates this kind of cool thing where when oxygen binds, it helps make the heme group adopt a shape, which then ultimately affects the shape of the protein, which then helps more oxygen to bind. And so you have this sort of like feedback loop of like oxygen binding causes a change in shape, which helps with more oxygen binding. Or when oxygen is not bound, it helps the protein adopt a different shape, which helps less oxygen be bound. So it's kind of this cycle where you have this cooperativity, right? Where you have it really wants to be in this kind of like all or none position where it's like nothing is bound, or like once something gets bound, it really wants to get more bound. And that is all related to all these different interactions we see, different types of charge-based interactions and covalent interactions and non-covalent interactions, all that biochem stuff. Any questions about T and R state? Is that a question?
Yeah, not about T or R state.
Okay.
It's about BPG.
Sure.
BPG is a result of cellular respiration, correct?
Yeah. So it's essentially it's what happens when you're breaking down nutrients and other food you eat, and you're breaking them down into pieces to ultimately form ATP. Yeah. And we'll go over that when we go over metabolism stuff a few chapters from now. Any other questions about this stuff? Cool. So let's talk a little bit more about the Bohr effect, which is related to that difference in T and R state at different pHs. So here we have 3 curves. They show oxygen binding by hemoglobin at 3 different pHs. So everybody take a few minutes, label which curve is which, decide which curve has the highest KD, and also which curve has the strongest oxygen binding. All right, so before we start answering these questions, let's just talk a little bit about what we know about pH's effect on the T and the R state for hemoglobin. So we just talked about how a high pH will stabilize which state? T or R? Yeah, R state. And does the R state have tighter or weaker binding? Tighter binding, yeah. So if we can answer this question, which curve has the strongest binding, we can figure out which curve is which based on pH, right? Because we know the tighter binding is going to be at the higher pH. So first, before we can answer number 3, let's actually first answer number 2. So which curve here has the highest KD? Yeah. The one all the way to the right. Yeah, the one on the right. Because we know if we have our 50% fractional saturation and we draw our horizontal line over, the one with the highest pressure requirement is going to be this one, the one all the way on the right. So what does that mean for number 3 now? So if we know which curve has the highest KD, the one on the right, that means the other ones have lower and lower KDs. So which one of those is going to have the strongest binding? What do we know about how KD relates to binding? Yeah, the left one, exactly. Lower KD, stronger binding. So, sorry if that's illegible. So if we have stronger binding, does that mean we're in the T state or the R state? Yeah.
Stronger binding.
Yeah.
R state.
R state, yeah. So that means if we have stronger, for question number 3, the stronger binding one, the one on the left, means we're in the R state. The one that has the highest KD as the weakest binding, it's going to be the most in the T state. And we already talked about when we started, the R state is going to be stabilized by the higher pH. So now we're ready to label our curves, right? So for our curve on the left, or actually let's start with our curve on the right, this one. What is the pH for that curve? Yeah, 7.2. Yeah, and what's the next one? 7.4. And what's the last one? 7.6. Any questions about that? Yeah.
So I'm like switching my log. When it comes to highest KD, that's regards to T state, and when it's O2 binding, that's the R state, correct?
Yeah, strongest binding, R state, which means lower KD. Yep. Any other questions? All right. Let's talk about fetal hemoglobin. So fetal hemoglobin, which is also called hemoglobin F, has different subunits. So we talked about, I think on our first question, how adult normal regular hemoglobin has 2 alpha subunits and 2 beta subunits. Fetal hemoglobin has 2 alpha subunits and 2 gamma subunits. And so it has this slightly different structure, and that means it cannot bind BPG. So take a few minutes and think about these 2 questions. So how would you expect the strength of fetal hemoglobin oxygen binding to be different from adult hemoglobin because of this change in BPG binding? And what is the physiological benefit to the fetus? Or like why does this help? So take a few minutes and think about that. Yeah, you have a question or do you want to answer?
I'm gonna try answering.
Okay, sure. So, how would you expect the strength of fetal hemoglobin oxygen binding to differ from adult hemoglobin?
Well, because BPG binds to the T state
Mm-hmm.
without it being the R state, so you have higher affinity.
Exactly, yeah. So there's no T state. And so if there's no T state versus R state, you essentially are always in the R state. So we have overall stronger binding. And there's also no cooperativity. And so you can think of fetal hemoglobin's binding curve as being more similar to the way myoglobin's binding curve looks, right? There's no cooperativity, and it overall binds to oxygen more strongly on average than adult hemoglobin. So what would this mean for the fetus? How does this help a fetus out? Yeah. Yeah, I mean, essentially, right? So think about the fetus's environment. Where is it getting its oxygen from? Is the fetus breathing oxygen into its lungs? No, right? So where is it getting its oxygen from? It's getting its oxygen from the mom's lungs, right? The mom is breathing in oxygen. There's oxygen on the hemoglobin on the mom's red blood cells. They share a blood supply, right? And so the fetus has to be able to then take oxygen from the maternal hemoglobin, and it does that because of this special fetal hemoglobin. So it acts in a way that's very similar to when we think about like myoglobin at our tissues in that low oxygen environment, hemoglobin hands off oxygen at the tissue. Similarly to how hemoglobin, the maternal adult hemoglobin, is going to be handing off oxygen to the fetal hemoglobin in relatively low oxygen environments, such as where the fetus is, right? The fetus can't get its own. It needs to basically steal the mother's oxygen, and it does that because of the special hemoglobin. Any questions about that? All right. So let's talk about sickle cell anemia. So everybody can take a few minutes and decide what they think is going on with sickle cell anemia, and we'll discuss it afterwards. Okay, so let's start talking about sickle cell anemia. So first we'll start with questions 1 and 2. So individuals with sickle cell anemia have a mutation in which gene? Yeah. Or do you want to go ahead? It's a beta chain. Yeah. Of which protein? Hemoglobin. This mutation replaces what kind of amino acid? Yeah. Yes, it is a charged amino acid. Do you remember which one? Glutamic acid, yeah. And where is it? Is it on the surface or in the core of the protein? On the surface, yeah. And what is it getting replaced with? Which is nonpolar. Yeah, so it's a nonpolar amino acid, and if we want to get specific, it is valine. So, and this causes a problem, right? Because the surface of our protein, like most proteins, generally has our hydrophilic residues, which impacts how water can help keep the protein in solution. Once you stick a nonpolar amino acid on the surface, then clearly we can wreak havoc, right? So what is happening here after this replacement? So question 6 and 7, this results in what? Yeah. Polymerization, exactly. So the proteins start sticking to each other, and ultimately, what kind of cells are dying? Red blood cells, yeah. Any questions about sickle cell anemia? Okay. In that case, we will move on to our last question. It's sort of open-ended. It's just explain this figure. So again, feel free to work with a neighbor on this. There's a lot going on in this figure. And maybe take 5 or 10 minutes on this, and we're getting towards the end of class, so we'll see. We might have to finish this one on Thursday, but anyway, you can get started on this, and I'll give you a few minutes to think about it.
That's probably a way for it to go back to the norm without taking out yeah, no, we can go. We can go I'm very like I sign on this.
All right, everybody. So let's start going over what's happening in this figure. So let's start here. We're breathing in oxygen at our lungs. There's a high oxygen pressure, about 100 torr. So what does that cause? What are we seeing happening? a lot of oxygen. Yeah,
Hemoglobin bonds to
exactly. Yeah. So at the lungs, we have hemoglobin. Hemoglobin is binding oxygen. So once we have hemoglobin starting to bind oxygen, we have some structural changes happening, right? So what state is the hemoglobin subunits going to be in? The R state. Yeah. So once we start binding oxygen, we have that shift occurring to the R state. Then our hemoglobin, it's carrying all this nice oxygen for our tissues, starts traveling through the blood. Eventually, we've arrived at the muscle. So muscle, very different environment, right? We have low oxygen pressure, about 20 torr. What else do we know about the environment at the muscle? Yeah,
I guess because of respiration, CO2 is increased.
exactly. So we're seeing respiration over here, right? So what does it mean to have respiration in our muscles? Like, are our muscles like breathing? Like, what does that mean?
Cellular respiration.
Exactly, yeah. So this is cellular respiration. And so ultimately, you can think of cellular respiration as it's a giant series of reactions that involves a whole lot of stuff that we'll talk about. But ultimately, you're taking oxygen and you're doing a lot of stuff with it. And ultimately, your byproduct is carbon dioxide. And there's a lot of stuff in between. But you can sort of think of that as like a very general overview of what's happening with respiration. So we're taking in oxygen in our muscle cells and in other cells, and we're releasing carbon dioxide while we're making energy that we're using to, for example, contract our muscles. So we have more carbon dioxide getting generated, and we talked about before, yeah, do you want to? Okay, and we also talked before about how during this process of making energy, we have more production of this metabolite that we discussed that stabilizes one of the states of hemoglobin. Does anybody know which one I'm getting at? Yeah, BPG. Yeah. So we have BPG over here in the muscle. So like and we also have a lower pH at the muscle, right? We have all this carbon dioxide, which helps promote a lower pH, like we talked about with all our blood buffer system stuff. So we have a low pH. We have low oxygen. So what does all of this mean for what our hemoglobin wants to do and what is so all this stuff, what does this mean for our hemoglobin? What state are we in? Yeah, the T state. Exactly. So we have our T state, which means we're releasing our oxygen. And what is taking up our oxygen? Yeah, myoglobin. Yeah, and so that's what we see on the side over here. So the hemoglobin is like, I'm tired of holding this oxygen. It lets it go, and myoglobin is very happy to then take up the oxygen and help hand it off to cells, which then will use it in the process of cellular respiration. And we also talked in the lecture, another thing that helps hemoglobin stay in the T state is that formation of carbamates, which is also enhanced by the presence of CO2. So we see that here too. So we have carbamates. So all this stuff is kind of helping the hemoglobin be in the T state. So at that point, we talked about how we have our cellular respiration and we're producing carbon dioxide. So what happens to the carbon dioxide? Where is it going? Or what's happening to it? Yeah.
more carbonate when there's more CO2.
Yes. Yeah. Okay, so we have carbon dioxide. It's getting made by our muscles. It's getting released into our blood, which we see here. Carbon dioxide going around this way in our cycle. And we have in our bloodstream, in some cells, we have this enzyme called carbonic anhydrase. Carbonic anhydrase will take carbon dioxide and water and help to make bicarb, or carbonic acid, right, which separates into bicarb and protons. So, which is, as we know, part of our blood buffer system. So, and so, yeah, so that now in this environment, right, we have a lot of free protons, which does what to the pH? Yeah, so we're still at this low pH environment, and so we're still helping to encourage our hemoglobin to be in which state? Yeah, our T state. Yeah, so we're still in our T state, and this is being helped out by the formation of our bicarb / carbonic acid. So we're still in our T state here. When we're looking at our hemoglobin, it's traveling back through the blood along with the carbon dioxide, which is now in the form of bicarb. Eventually we get towards the lungs again, and now a couple of things are happening, right? So now we have a change in the local environment at the lungs. So in our muscles, we're at this really like low pH kind of state. In our lungs, we have a much higher pH. And so part of how we can kind of get this to happen is we also have carbonic anhydrase. So over here near the muscle, we saw carbonic anhydrase helping to convert carbon dioxide as a gas into bicarb, which helps it to travel through the blood and not be a gas. Once we're at the lungs, we're ready to get rid of our carbon dioxide. So once again, we use carbonic anhydrase, same enzyme, other direction, and now we turn our carbonic acid back into carbon dioxide, releasing a water. And now we have carbon dioxide gas that we can breathe out in our lungs. And now, since we're expelling carbon dioxide, we have this higher pH environment. And so what does that do now to our hemoglobin in this higher pH environment that's full of oxygen? Exactly. Now the hemoglobin wants to go back in the R state. It wants to pick up some more oxygen and go through this whole cycle all over again. Does this make sense? Any questions about this cycle? Yeah.
Once the CO2 is the hemoglobin will want to go back to the oxygen.
Yeah, so it's a higher pH environment, right? Because we're consuming these protons by turning with carbonic anhydrase by making carbon dioxide gas again. So that itself is helping to raise the pH. And we know that a higher pH will help to stabilize the R state of hemoglobin. And we also know that when there's more oxygen, which you have in the lungs, hemoglobin will want to take up more oxygen. And so ultimately, between all the different factors that are present there, you're helping, hemoglobin start to adopt the R state again.
And that's the example of graph.
Yeah. Any other questions about oxygen transportation and storage? Yeah. Yeah, that's a good question. I guess, yeah, I mean, at some point between when it shifts from the T to the R state, which is not an answer to your question, it's not really shown in this chart. That is also a good question. I will look into that. Yeah.
So carbonic anhydrase or one of its functions is to convert bicarb back to CO2.
Yeah, so it does both ways. So it makes CO2 into carbonic acid, and then which separates into bicarb and protons. And then when you get to the lungs, it reverses it. It takes the protons and the bicarb / carbonic acid and turns them into CO2 again. Yeah, same enzyme. It does both directions. Yeah. Okay.
Is it interchangeable? You use bicarb or carbonic acid, or does it turn it into one of those and they use that?
Well, it's the same thing, right? So bicarb. So this is carbonic acid,
Right.
And then when you lose a proton, it's bicarb plus proton, right? So depending on the exact pH, we'll decide whether or not it's dissociated, right?
But carbonic acid can flush through both.
It,
I mean,
Yeah.
like in hydrate.
yeah, I mean, in this diagram, it's showing that it's taking bicarbon protons, which is, I think, what you would see in this pH environment. I think it probably can also take up carbonic acid if it was at a different pH, but at this particular pH, we see it this way.
Okay.
Yeah. All right, so yeah, that's our time today. So just a couple reminders. So tonight, your paraphrasing responses are due. So hopefully you've either done that or you're going to do it before midnight tonight. And also a reminder, quiz 6, I believe, or 5. Quiz 5 is due on Thursday. Yeah. And that's about chapter 6, which is why I'm confused. So yeah, I'll see you guys on Thursday.